Good morning, everyone. It's Dr. Liberty Bonastro. Good morning. Today, I have the pleasure of introducing Dr. Robert Freund to present the first grand rounds of 2024. Dr. Freund received his undergraduate degree from the University of Michigan, graduating with distinction with a degree in business administration and a minor in biochemistry. He then went on to work at the laboratory IT company SunQuest before attending medical school at Drexel University College of Medicine in Philadelphia. He joined the University of Minnesota as an anatomic and clinical pathology resident in July 2020. During his time in our department, Dr. Freund has published one article in the Journal of Neuropathology and Experimental Neurology and presented abstracts at USCAP, ASCP, and ACG conferences. He is a current junior member of the CAP Council on Informatics and Pathology Innovation. Following completion of his residency, Dr. Freund will be leaving for Portland to attend the Oregon Health and Science University, where he will complete a gastrointestinal, pancreatobiliary, and liver pathology fellowship. It has honestly been my absolute pleasure working with Dr. Freund as a colleague during our residency. And today he will be presenting regulation of laboratory developed tests, implications of the U.S. Food and Drug Administration's new proposal. Please join me in welcoming Dr. Robert Freund to Grand Rounds.
Thank you for the kind introduction, Liberty. So as she said, we're going to talk about the regulation of lab-developed tests. So we're going to define lab-developed tests, review the history of their regulation. We're going to discuss the FDA's proposed rule of medical devices laboratory-developed tests that they published in October. We're going to describe the implications and potential impact on medical labs if the proposed rule is adopted. So what is an LDT? LDT stands for lab developed tests. There's no legal definition for a laboratory developed test, but the FDA is generally considered an LDT to be an in vitro diagnostic product that is intended for clinical use, that is designed, manufactured, and used within a single lab, and that lab needs to be CLIA certified to perform high complexity testing. There's a lot of items in this, and we will sort of go over them as this presentation continues. But I'd like to start with, who is the FDA? And so our story starts in 1837. Henry Ellsworth, he's the commissioner for patents at the U.S. Patent Office, and he sends a letter to Congress and says, we have a lot of entrepreneurs, and they are trying to patent some new seeds and plant varieties. And so Congress sends $1,000 to the Patent Office for agricultural purposes. Within the next decade, they've established a chemical lab within the agricultural division at the U.S. Patent Office. And in 1862, Abraham Lincoln establishes the USDA, the Department of Agriculture, and appoints this guy, Charles Wetherill, to head that chemical division. Around this time, America has a problem with adulterated food. There's like sawdust in the flour. There's formaldehyde in the milk. If it's cheap and it kind of looks like food, it is being sold, and inedible, it's being sold as food. 1906, Upton Sinclair publishes The Jungle. He means it to forward the socialist movement, but what Americans take from it is not that the working conditions in the factories are poor, but rather that their food is unsanitary. Pressured by the new head of the chemical division at the USDA, Henry Wiley, Theodore Roosevelt is meant to signs the Pure Food and Drug Act of 1906. And this is the sort of start of the FDA. The Pure Food and Drug Act prevents the manufacture, sale, and transport of adulterated or misbranded foods, drugs, medicines, liquors. And then in 1937, company Mesangial is making this drug sulfanilamide. And the way they do it is they're basically dissolving it in the equivalent of antifreeze. 100 patients die, and the public is understandably outraged. And Franklin D. Roosevelt ends up signing the Federal Food, Drug, and Cosmetic Act, or FD&C Act of 1938. This again prohibits adulteration of food and drugs, but they add on medical devices and cosmetics. It also gives a bit more regulatory teeth. They can do injunctions and so forth. Manufacturers are now required to test and prove that their drugs are safe before they're released to public. And it's sort of the first concept of pre-market approval. And then I wanted to take a look. It defines devices and it defines drugs. But the language that they use for defining devices and drugs is very broad and very similar. So if you look at the blue and you look at the blue here, it's almost the same language, same for red. And this comes into play in 1968, where the FDA tries to regulate an antimicrobial susceptibility disc as a drug, and the court agrees. Basically, they say that that language in that previous term, the FDC Act, was so broad that the term drug does not necessarily just refer to a drug. The word drug is a term of art, and it sort of encompasses all those definitions. And so they gain this ability to regulate a device as a drug. Then in 1945, Harry S. Truman, he proposes universal health care. Unfortunately, we have our second Red Scare. James McCarthy is in Congress, and the American people are afraid of communism and socialism. We do not get it at this time. But in 1965, President Lyndon Johnson signs Medicare and Medicaid. And so we got Johnson, we got Harry Truman over here on the right, and we get Medicare and Medicaid, which is covering seniors and low-income Americans. And so with new federal progress comes new federal regulation, and President Johnson subsequently signs the Clinical Laboratory Improvement Act of 1967. It's part of a larger thing, the Partnership for Health Amendments. And it establishes licensing requirements for laboratories operating across state lines. And basically, they're doing this to stop people from defrauding the government. So they set up some minimum quality requirements for laboratories. We have our next great scandal in the 1970s. So a company, A.H. Robbins, produces this Delkin shield. It's essentially an IUD, and it ends up causing infections, pelvic inflammatory disease, stillbirths, infertility, deaths. And again, the public is sort of outraged. President Ford signs the Medical Device Amendments Act of 1976. And in this, it redefines a device. And again, the language here, it's very broad. I want you to focus in on this in vitro reagent. In the future, the FDA is going to use this definition to argue that it has authority over lab developed tests, and sort of based on the past court's decisions on the all-encompassing broad language, they're probably right. How a new court would rule is who knows. The MDA 1976 Act also adds a risk-based classification system for medical devices. So class 1, this is going to be your low risk. Class 2, moderate risk. Class 3, high risk. And your moderate risk is going to require pre-market notification with a 510K clearance. Your class 3 your high risk is going to require actual pre-market approval. We have our next scandal in the cut rate pap mills that cytology is familiar with. And again, so there's these poorly functioning labs. They're missing cervical cancers through laboratory errors. And again, American public's outraged. And President Reagan signs the Clinical Laboratory Improvement Acts of 1988. And it revises and improves the oversight and certification of clinical laboratories. So they issue some standards, quality assurance and quality control program. This is modified over the years. So 1990, they add written protocol and documentation, method validation. 1992 is they're actually sort of referring to a lab developed test here. Basically, they're saying you developed in-house, a test not cleared by the FDA. And at this point in time, so 1977 to 1992, the FDA hasn't really said anything about lab developed tests. They haven't said anything about these types of things. And now they have. So 1992, they issue this draft compliance policy guide, basically saying it's come to their attention that laboratories are manufacturing homebrew products, and they think that these things fall under the same regulatory requirements as an unapproved medical device. Now, this is a draft compliance. It doesn't actually mean anything. It's not law. But the point is, the FDA has now noticed. And so during this time frame or after this time frame, there's many other proposals and discussions and documents and talks, but they haven't, for the most part, are not signed into law. And so they haven't made this timeline here. There are some things that have made it on the timeline that are kind of minor, that don't add to the larger discussion today, but they are law. So I've included them. So Clinton FDA or the FDA Modernization Act of 1997 amends the definition of a wave test criteria. We'll go over what the different types of tests are as this presentation continues. 2003, the CDC and CMS add specific requirements for lab developed tests. Again, they're not using the term LDT, but they are saying this modifies an FDA system or uses a non-FDA approved system. Obama signs the Test Act, basically says when you're doing proficiency testing, you can't send your sample off to another laboratory for analysis. And then in 2014, the FDA drafts another document, basically suggesting that labs should do pre-market review and post-market surveillance, not requiring it, but recommending it. 2016, that document is withdrawn. Around this time, the public gets notice or starts to realize that something's going on here. So is lab testing the Wild West of medicine? Largely unregulated industry comes under FDA scrutiny. Lab developed tests providers fight back. And that is what happens over subsequent years. In 2018, the first version of the VALID Act, or the Verifying Accurate Leading Edge IVCT, In Vitro Clinical Tests Development Act, is written, and it is proposed to Congress or introduced to Congress. And from 2018 to 2023, it continues to be revised and sent to Congress. The VALID Act has a lot of similarities to the new FDA's proposal, so I'm not going to go over the details of it now, just to say that this is not necessarily a new topic. This has been discussed for many years now. And so late September, the FDA announces that they are proposing this rule for lab developed tests. They subsequently publish it on October 3rd. And then they host a webinar to sort of explain their logic. So back to this definition, what is an LDT? Generally, in vitro diagnostic product intended for clinical use, designed and manufactured, used within a single lab that's CLIA certified to perform high complexity testing. I want to focus in on the word manufactured for a second. Basically, for a lot of people or a lot of labs developing lab developed tests, their logic is, well, we're not actually manufacturing anything. We are just, we're assembling components. We're taking things that are already approved. We're just modifying things a little bit. And the FDA, like those definitions of device and drug, they intend for manufacture to be very broad. And so they're including design, preparation, propagation, assembly, processing, basically anything. If you are making it, it's manufactured in their eyes. Mostly for the residents, I wanted to go over the different types of clinical laboratory tests. So there's 3 types of tests. And so we have wave tests, and then we have non-wave tests. And the non-wave tests can be moderate complexity tests or high complexity tests. And how do you know where your test sort of fits in? You can go to the CDC and they have lists of tests, and you can see how complex is your test. And how do they decide? They have a scoring card. We're not going to go over this whole table here, but they have a card, and it has items like knowledge and training and reagents and characteristics and interpretation, and it scores each item and decides whether it's high or low complexity or is eligible for waiving. These are your CMS approved CLIA accreditation organizations. So organizations like CAP or the Joint Commission. There are 2 states which have CLIA exempt labs, and so that's Washington and New York. And basically, CMS has said that their lab requirements are more stringent than CLIA. So there's a couple different certificate types. So certificate of waiver, this allows you to perform those wave tests. Those are sort of basic tests that you can perform at home and that are low risk. The risk of something bad happening is very low. Certificate for provider performed microscopy. Those are those tests you might, a clinician might do sort of in the patient room or, the hallway. Certificate of registration, compliance and accreditation. These allow you to actually perform the high complexity testing. Registration basically says that you have announced that you want to be doing high complexity testing. Compliance means your lab has been inspected and it is in compliance. And accreditation is you've actually gone through all that and have been issued your accreditation. Here's ours. And then here's a couple individual ones for some of our labs on East Bank. So what is the FDA proposing? So this is the slide. This is sort of the big summary of the FDA's October proposal. They are proposing to amend its regulations to make explicit that IVDs, in vitro diagnostics, are devices under the Federal Food, Drug, and Cosmetic Act, the FD&C Act. And basically, they claim that it's already been this case. We're just writing it down so you guys definitely understand. So what they're saying is they intend to phase out their general enforcement discretion for these lab developed tests. So they say, we've always had the authority to regulate these lab developed tests, and now we are exercising that authority. So the way the FDA sees it is there's two regulatory pathways. There's the commercially distributed test pathway and the LDT pathway. So commercially distributed, you have a test kit. It's approved by the FDA, and then it's distributed out to patients, hospitals, clinical labs, and then goes to the patient. And then there's the LDT pathway, where the test is made in a single lab, and then the FDA says they're enforcing sort of discretion. They have the ability to regulate this, but they haven't been doing it. And then those LDTs sort of enter the market without any review before going to the patient. So they say that there's sort of gaps in that LDT pathway. In the CLIA pathway, they say there's no research phase, that analytical validation, they're only using post-hoc sampling, there's no clinical validation, that adverse events are not being reported out, and that the results are not really transparent. The public doesn't really get a list of things. They don't really know what's going on with these LDTs. I have this table here to give a sense of how the FDA is classifying in vitro diagnostics, what kind of test would be low risk versus high risk. So your class 1 low risk test would be something like a blood glucose meter. It would be sort of exempt on this FDA pathway, and so they just have sort of basic general oversight. Class 2, your moderate risk, that's more like an immunoassay, and you would have to get this 510K pre-market like notification type thing. And then for class 3, your high risk, that's something like next-gen sequencing, and you would need pre-market approval before you could use this sort of LDT. So again, the FDA claims enforcement over all LTDs, and they intend to exercise that enforcement. They do say that they are going to have sort of enforcement discretion for certain types of tests. So your pre-1976 tests, your manual tests, they're going to exercise that enforcement discretion, HLA tests, forensic tests, and public health surveillance. They have a proposed timeline in which to sort of put forth this process. So about 3.5 years out would be when you would need pre-market review for those high-risk in vitro diagnostics. So the FDA has a few justifications. One of their main justifications is a series of 20 case studies that they published in 2015. They also discuss some more recent evidence, which for purposes of time, I'm not going to go into, but they add in some other items here. Some of them, they're a little sparse on actually saying what they are. So, you know, they say allegations of problematic tests, but they don't necessarily list them beyond, you know, the past tests, the past 20 cases. We're going to take a look we're going to take a high overview look of the 20 cases. So the FDA thinks that they need oversight of LDTs to address a few concerns or serious concerns. They say there's deficient adverse event reporting, there's no pre-market review of performance data, there's unsupported manufacturer claims, inadequate product labeling, lack of transparency, an uneven playing field, threats to the scientific integrity of clinical trials. They say that these LDTs are often being performed without informed consent in the clinical study, and that there's no comprehensive listing of all the LDTs currently being used. So here's their 20 case studies, and they've sort of grouped them into big categories. So false positives, false negatives, both false positives and false negatives, no clear relevance to the disease, disproven scientific concepts, undetermined or undermined drug approval or drug treatment selection, and their catch-all other unvalidated tests. We're not going to go through all these, but most of these are legitimate, and some of them are sort of big cases that you may have heard about in the news at sort of well-respected institutions. So we have this Duke case where they were running a proteomic microassay to sort of group people in a clinical trial, and the assay was faulty, and it was kind of a scandal. The report argues basically that CLIA is not enough, and all these problematic tests were offered at laboratories that were following the minimum requirements for CLIA. And then another big public case is Theranos. This is where they could, run all sorts of laboratory tests off just a little tiny drop of blood, which turned out to be not true for the most part. Funny enough, they did have one assay that went through the FDA and was approved to be equivalent. Whether it's a good assay or not, I don't know, but this herpes simplex virus assay did pass the FDA. For the most part, though, they were running assays off of, so they were running FDA-approved assays, but at dilutions that were not approved by the FDA, and those dilutions they were using as LDTs. This was actually caught, though, by a combination of investigative journalism and CLIA lab inspections. So there's an argument that CLIA was doing exactly what it should do. The FDA lists some potential exemptions right away. They say they're going to, or they list some exemptions and say, we need to discuss them, whether we will or will not include these. But they acknowledge that these items exist. And so they say there should be enforcement discretion, or should there be enforcement discretion for academic medical centers, which would be good for us here. That said, you have to very carefully define academic medical center. In the past, they've called an academic medical center sort of one address, one location. And we have at least 3 locations, more if you count all our sort of ancillary sites. So something that will have to be defined carefully. Grandfathering. So they say, well, maybe we say all currently issued in vitro diagnostics could be offered as LTDs. It could be good, again, for us, but is that necessarily good for the public? I'm not so sure. They say maybe small labs need a longer phase out period. It might be difficult if we suddenly take away all their LDTs. And then they say we might need discretion for public health scenarios or vulnerable populations. So basically, to implement this rule, we would need many more rules and guidance documents. No specifics have really been mentioned yet. This is just the FDA saying we have the authority and we're going to do something. So they asked for public comments, especially on those items of discretion, but on sort of any thoughts that the public had on lab developed tests. And they got, this is now closed. Otherwise, I would encourage you to go submit your own comments. So they got 6,707. They're public, you can peruse them. I like this one from the Broad Institute, the Harvard Associated Broad Institute. They say the proposed rule is a sledgehammer to crack a nut. ARUP has a nice letter as well, but they've got this nice PowerPoint slide basically, which groups their complaints, statutory authority over LDTs, regulatory impact analysis, general concerns. I largely agree with these regulatory and general concerns that they have. The statutory ones, for my readings, I think the FDA has a fairly good argument with sort of the old court system that, at least in the law, these LTDs probably are devices. Interstate commerce, while sort of and commercial distribution, sort of in the very narrow view, it might seem that labs could sort of be operating in their own state and not, the device manufactured in your personal lab is only for, your patients in the small area. It's not going outside. In the past, the courts have ruled that if the reagent is crossing state lines, or if there is economic benefit that is large enough, that counts as interstate commerce, and basically regulation would still apply. But these are topics that people have opinions on both sides, and so I think it is definitely worthy of discussion. So are LTDs a product or a service? So the FDA is more or less thinking of them as products. You build something, you lock it down, you never change it. Whereas a lot of labs are thinking of tests as a service. They're not assembled in kits. They're not physically distributed. You have processes, procedures, protocols. There's a board certified pathologist, and they're providing an interpretive element with expert medical opinion. We have these interpretive comments. The tests are more dynamic. Sometimes tests break and you need to fix them. So this is a study out of ARUP, and they wanted to look at, okay, how many tests are we actually doing? How many LDTs are we actually doing at our lab? And so they say that the number of LTDs they're doing is actually relatively a small percentage of all their tests. They say it's only 3.9% of all their tests. That said, they have 880 or 45% of their distinct assays are LDTs. So they have in my opinion, sort of a significant number of, distinct assays they're using. And then they say of just like modified FDA tests, they have 49. Now that said, those numbers are probably an underestimate. They've defined a modified FDA approved test pretty narrowly in their paper as just a change in a specimen type, preservative, or collection device. But any modification to a test may make it an LTD. So if you extend the clinical reportable range of your assay, increase the dilution fold, that's basically an LTD. If you modify the thresholds for tolerance for an interference substance, that's going to be an LTD. If you change the intended use, that's an LTD. For this study out of ARUP, their most frequently ordered lab developed test was tacrolimus. You'll notice here that a lot of these are mass spectrometry tests. And in their study, mass spectrometry is the most common and most frequently ordered LTD. Now, mass spec, those are actually class 1 devices, low risk, but a lot of the analytes are class 2 moderate risk. So they'd have to get more clearance. I want to talk about pediatric reference intervals. Most FDA cleared assays do not have pediatric reference intervals. They're expensive, so manufacturers don't do it. Laboratories frequently regulate their own reference intervals. Pediatrics are not just little adults. You have to modify these assays. You have to change the intended use. If pediatrics isn't on the packet insert, you have an LTD. Newborn screening, almost all of those are LTDs. For rare disorders, frequently pediatrics has many rare disorders. There's no other tests available. The FDA doesn't have tests. For toxicology, a lot of the screening assays, there are FDA approved immunoassays, but almost all the confirmatory tests are LDTs. So mass spec, all your therapeutic drug monitoring, drug testing. I saw someone put in the argument that, we're in an opioid epidemic and you can't be doing this in an opioid epidemic. And, maybe it's a fair point, but it's just one in many broader arguments. Pharmaco, I want to talk about pharmacogenetics. So there's a genetic basis of different metabolites, and in these cases, interpretation of tests results is very critical. And you have to do a lot of customization for your patient population. It's going to be very difficult to sort of regulate this as a medical device. I have a couple case studies here. So this is a study out of Dartmouth. They said that during the COVID-19 pandemic, they had staffing constraints and supply chain challenges that sort of required them to send all their mass spec out. So normally they do mass spec as an in-house LDT, and they had to send it out. And they said that this would be analogous to the loss of LTDs if they're regulated as the FDA is proposing. And so the study analyzed the effect on therapeutic drug monitoring and opioid testing panels. Specifically, they look at turnaround time and test costs. So here we're looking at the turnaround time for cyclosporin A, sirolimus, and tacrolimus. So it's like OA, siro, tacro. On your vertical column here, we have time. So black is going to be your in-house tests. White is going to be your send-out tests. And you can see for cyclosporin A and sirolimus, the send-out took about a day longer. So 24 hours longer for those two tests. The tacrolimus turnaround time was unchanged. They said there was a clinical impact, or the physicians were complaining, basically. The delayed reporting of cyclosporin A sort of complicated the initial dosing of pediatric patients on the hematology service. They also look at financial impact. They say the cost of performing a test was more, 3 times for sirolimus, 2 times for verolimus, 2.5 times for opioid screening. I'll say they didn't include labor costs, which maybe they should have. They did this sort of rough cost for opioid testing. They said, okay, there's a reduced cost to the lab, but then we have a send-out cost, and then there is an anticipated reimbursement loss, and that gives you a total cost of $554,000 a year. There will be additional costs to the lab for sort of just the approval process of a medical device. So these are the medical device fees for 2024. There was an almost 10% increase from 2023, funny enough. If you have a high-risk test, sort of your class 3 medical device, that's going to cost at least $483,560 per test. Medium risk test, that's only $21,700. If you have a high risk test, every year you have to pay this reporting fee, so that's another almost $17,000. And then there is, of course, an annual registration fee, which is $7,600. So for your low volume, low margin tests, that might not be financially viable for many labs, especially your small laboratories. And then for your high volume LTDs, that could also prove to be expensive. In the Medical Device Amendments Act of 1997, there is language saying that the FDA shouldn't impose requirements that are unduly burdensome, and that they need to take into account the cost of complying and the need for protection of the public health. So this argument or this law might come into play in this discussion, whether these costs are prohibitive or not. I have another case study here looking at mpox testing. So the US declared a health emergency for mpox. And the FDA sent out this notice saying that the FDA does not intend to object to the use of monkeypox serology tests for high complexity CLIA certified labs. And specifically, they mention academic medical centers. In this document, they say that that has some certain requirements. One of the requirements is that tests use lesional swabs. At UCLA, they were already bringing up an LTD for mpox when the public health emergency was declared. They notified the FDA that they had this test, and the FDA sent notice to UCLA that that's great, but you need to suspend it for non-lesional specimens. At this time, UCLA had a high-risk patient present to UCLA without any lesions, and they did a test for mpox that was positive on a rectal swab. And they wanted to continue doing these rectal swab tests. And so they submitted to the FDA an emergency use authorization. And ultimately, this request or the emergency use authorization was denied by the FDA. And they deemed that the testing was unsatisfactory. This paper goes on to compare the CLIA requirements to the FDA requirements for this emergency use authorization as kind of a comparison of what the new regulation will look like. So under CLIA, you have to show that your test has certain performance characteristics, accuracy, precision, analyte, sensitivity, specificity, reportable range, reference intervals, anything else that's important. This paper has a big table of comparison. We're not going to go over that due to time constraints. But a couple of items that UCLA thought were sort of more stringent and caused issues for them were the FDA was requiring a multi-lot study. UCLA only wanted to do a single lot because they had a supply shortage and there was some lot-to-lot variation. And so it was causing some issues. The next item is sample stability. The FDA wanted at least 50 samples at each time and temperature condition for sort of real world conditions. And it required UCLA to sort of make a bunch of contrived testing samples, and they had a dedicated courier, and basically they had to spend a lot of money on some workflow adjustments to sort of get all the data that the FDA wanted. And then the FDA wanted a prospective blinded randomized study, with an IRB protocol, which was, time consuming. That said, UCLA did all these things, but they were sort of unable to actually substantiate directly through in-house study the results. The literature, they argued, should have been good enough to show that this was kind of a working test. They say the performance of their rectal swab didn't improve following these processes, nor did they notice any sort of deficiencies. That said, sort of the number of cases in sort of the county were decreasing, and so when they got pushback from the FDA, they decided rather than to continue to pursue this, they would just withdraw their application. So they kind of argued that the FDA requirements were kind of burdensome, but I think there's also an argument here that maybe the FDA is correct, and maybe they, prevented a test going out that is not necessarily valid. I'm not going to sort of go into that further, but I think it's something to think about. I want to talk about manual testing. So manual tests are exempt, or the FDA is saying they're exercising this discretion of certain regulatory requirements. only if they're considered, not high risk and no component or reagent is introduced into interstate commerce. And I'll say, based on sort of this broad definition of interstate commerce, probably almost every reagent goes through interstate commerce, in that it has a large economic impact and/or, is going to cross the state line at some time. That said, pathologist interpretation of immunohistochemistry or in situ hybridization may qualify as manual testing. So IHC, it's a pre-1976 method of testing. The FDA argues that manual testing is safer than automated testing. Many laboratories disagree. How much IHC is actually being performed manually versus an automated method? I think a lot is being performed automated these days. And then the manufacturers of reagents used for manual tests may be subject to new regulatory oversights. So the lab itself might not be, but there might be new regulation on the manufacturers of these reagents. I have another case here. So here we're looking at, we're comparing an FDA-approved HER2 evaluation kit versus an LTD. And here we have it's breast cancer, and here is the FDA-approved HER2 stain, and here is the LTD-approved stain. And what this study ends up finding is that the LTD showed higher sensitivity than the FDA-approved kit, 100% for the LTD versus 77.3% for the FDA-approved kit. Their gold standard here was FISH. Basically, they compared the results for the FDA kit and for the LTD against FISH. And the LTD classified all of their cases as, +3 or +2, whereas the FDA kit classified some of the positive cases, 5 of the positive cases, as +1, which would have been a miss in this study. So CLIA and CAP, they're not going to go away. Performance standards of laboratories might change. A new standard might be placed on top of CLIA. But the FDA is going to finalize some sort of proposal. They have to respond to the comments. They're going to group them by categories. The timeline's not exactly known yet. I've heard April, I've heard June, but who knows? A lawsuit against the FDA is probable. In that situation, would the court grant a preliminary injunction? I don't think we can say yet. So would a court say, basically, don't advance this rule? And then if they do grant an injunction, should your lab comply or not comply with the rules? And the question ends up being, how risk adverse is your lab? Go talk to your compliance officer. How is this going to affect us? We can't really say yet. Regulation is going to be most worrying for these small hospital labs in the small communities. The FDA has already sort of said that they're maybe thinking about some sort of exemption for academic medical centers. Although how they do that is going to be very critical, how they define an academic medical center. Cost of compliance will likely increase, but I think this is still sort of a wait and see game. And so I have many references here, but that is my presentation. Thank you for your time and attention, and I'll take any questions.
Quick question here. Thank you very much, Robert. That was really very nice, especially the historical context that you started with. I appreciate that. Question, usually when FDA does this, there's usually an inciting event, and I'm not sure if you covered that. So what started all these LDT new rules? Do we know?
So I think it's sort of been a long building process. I think, some of the examples are like Theranos. that was 2015. But even before that, if we go to like those 20 case studies, I didn't go through there, sorry. I did not go through all these case studies, but a lot of these were actually higher profile cases. I think genetic testing was kind of an issue. Things like 23andMe were sort of releasing tests that were not FDA approved, and FDA thought, okay, that's a little bit too far. So I think that is partially the inciting event. But really, it's a long time building. Another thing that I didn't really talk about was during the COVID-19 pandemic, they issued emergency use authorization, and a lot of bad homebrew kits were going out. Basically, COVID-19 testing kits that didn't really work. And so that might also be an instigating event for this October law.
And I see John's answer. Just quickly follow up. I don't usually support FDA's regulation. I think sometimes, well, most times they tend to be like an overreach in everything. But at the same time, I think there has to be a lot of discussion about what is the role of the commercial labs in this. And I suspect that some of the problems that are coming up is now because of the commercial labs issues. I attended one of the CAP webinars on this topic last year, and that was one of my impressions. So I think going forward, the importance of being able to define what is academic versus not academic is probably going to be the critical role, because I can't say FDA should answer completely. I think they have some role to play, in this at the end of the day. But thanks.
Thank you.
Hey, Robert, Tony, that was a great presentation and very timely. A couple of comments. One is, I think the expected time frame for the FDA to issue its final rule will be not later than early May, and the reason for that is that within the federal government, there's a sort of policy that agencies don't release major rules within 6 months of a major election, when we have elections coming in November, because a new administration may come in and change the whole thing. So you can't sort of put in regulations the week before you leave office.
That makes sense.
I mean, there are many other aspects of this. One is the FDA does not have the staff to review and process all of these applications. And there will certainly be a lawsuit when they release the final regulation, because most medical scientific associations are completely opposed to granting FDA additional authority over this area. And there's also a lot of political stuff that's been going on too, that the philosophy is, do you let the FDA, a government agency, sort of make this big land grab? Excuse me. And there was an editorial in the Wall Street Journal calling it a land grab back maybe in January of this year. So there's a lot of political aspects to this too. And the Valid Act was turned down by the Congress last year in December of 22. And so for the FDA to sort of override, well, not override, but sort of to ignore the fact that the Congress didn't give them authority under Valid, and then just write regulations anyway, sort of is an odd situation, to put it, favorably. So I don't know what's going to happen, but there'll certainly be a lawsuit come this summer.
I'll speak up. Robert, I enjoyed the talk very much, and particularly the historical perspective. Reminded me of two things. Number one, when I first started setting up anti-nuclear antibody studies back in the early 1970s, I realized that I never would be able to do that now, but I sure learned a lot about anti-nuclear antibody studies by setting them up. But the other comment I wanted to make was your comment about Johnson signing Medicare and Medicaid into law in 1965. Johnson never meant that to be limited to seniors and people in lower income brackets. His idea was that eventually that would become a universal single payer health care. And of course, we are not there yet. However, I just want to point out Minnesota is very close, and we very well may at least have a vote on this by 2027. And so I just want to alert people to stay tuned to that.
Thanks, John. And I just wanted to kind of build on Tony. People saw me post this too, but I mean, I think especially for the trainees and anybody who's been dealing with the VALID Act and other stuff over time and has submitted, whether it's emergency use authorizations or other things to the FDA, I think it's critical that everybody in medicine understand that the FDA is predominantly a legal and financial slash, commerce agency. It is secondarily looking over aspects of science and medicine, but what drives most of their procedures, most of their thought process is legality. And so I think that that's something that we should all keep in mind. So it's not necessarily a bastion of scientific knowledge. It is a bastion of legal process. And the one comment that I put in the chat, and again, this is something that I released too when the VALID Act was coming out to just try and keep it as clean and clear as possible. All of the data that has come out so far has shown in general that LDTs have exactly the same or better performance characteristics than in vitro diagnostics. There's one study that came out which highly skewed the process, and which the CAP actually forthcoming will have a nice paper that clearly shows that that was a skewed and biased evaluation of the performance of LDTs in comparison with in vitro diagnostic approved kits. And so, I mean, the only thing that this is going to do is it is going to increase costs. It is going to decrease test access. I personally was part of an effort that the University of Minnesota and the Masonic Cancer Center did with ACS last year to get biomarker coverage pushed through all at least public insurance holders in the state of Minnesota. And laws like the VALID Act will decrease access to testing. And this is a very, very bad thing for patients.
Thank you very much. Congratulations, Robert. You are done.
Thank you.